Hello, good morning. So this is going to be a third lecture. Unfortunately, and when you say fortunately, we couldn't do this because of your practical classes. So I deemed it wise to record this online. So we did time. And hopefully on Wednesday, I'm going to come in and conclude my part of this course. So we're going to be talking about monoclonal antibodies today. Um, the team is with biotechnology. I think we have a lot of students from different backgrounds. So some of you did not study maybe biochemistry, biological sciences, microbiology, or any life sciences related course in your bachelor's phase. But I want to still believe you know what an antibody is. Antibody is a protein used by the immune system to identify and neutralize foreign objects, basically antigens, pathogenic bacteria, viruses, what have you, right? So we have two types of antibodies. We have the monoclonal antibodies and the polyclonal antibodies. From the name mono, poly, one, many, right? Monoclonal antibodies are antibodies that are identical because they are produced by one type of immune cell. So they are all clones of a single parent cell, while polyclonal antibodies are antibodies that are derived from different cell lines. So basically, when you talk about antibodies, you can't talk about antibodies without talking about the immunoglobulins, right? We have immunoglobulin G, which is very popular, immunoglobulin A, M, D, and immunoglobulin E. So according to differences in their heavy chain constant domains. Now, this is pure biochemistry. We're going to talk about this, I think, in the upcoming slide. So I've already defined a monoclonal antibody. You can just move forward. You can read this slide on your own. So basically, this is what an antibody looks like. It has more of a Y shape. And there's an antigen binding sites because they bind antigens, they neutralize antigens in the system. So they should have an antigen binding site. So structurally, you have to look at the chains. If you still remember your protein biochemistry, protein amino acid biochemistry. So, antibodies mostly are proteins. So there's a heavy chain, which is this below, and there's a light chain, which is here. I have a better figure in the next slide, don't worry. All right. This is just an overview, a sketch, give you an idea of what an antibody looks like. So I'm taking my time because of, it is a multidisciplinary course. We have people that didn't study anything like this, probably. Yeah, bachelor's degree this. All right, so the variable region of an antibody binds to the antigen. Okay, so this is the variable region up here. This is your variable region, and then this below is your constant region. There's a variable light chain VL, constant light chain here, then the constant heavy chain in the constant region. So there's also a classification of the antibody we call FAB region. This is the antibody fragments, antibody binding fragments, antibody binding fragments. It makes sense, right? It makes sense since this is the region that binds the antigen, antibody binding fragment. And the FC stands for the crystallizable fragments. I think it's here, right? Okay, so, and then this constant region here, the FC region, is made up of domains. This is a domain, this rectangular looking shape you're seeing here is called a domain. You still remember a protein biochemistry, you should know what I'm talking about. Or if you don't, you can still browse, right? Look for any biochemistry test. The homodimer, heterodimer. I don't want to go deep into that, but at least you get what I'm trying to say. So you have 1, 2, 3, 4, 4 domains there, right? This constant FC region. And this is to intimate you with the terminologies in this area of biotechnology. 4 FC regions. I've shown you the FC region here already. We have 1, 2, 3, 4, right? 6 FC regions. This is the constant region, right? The crystallizable fragments. 1, 2, 3, 4, 5, 6, right? So antibody drug conjugate. So this is the drug. This has a lot of spike around it. So you have each drug binding to a domain of the FC region, right? 2, 3, 4. And this variable region up here is known to bind antigens, the chimeric, right? What is a chimera? What is a chimera? You should know what that is. You can browse. It shouldn't give you everything, right? Humanized antibody. What is a humanized antibody? Bispecific antibody. These are things I might ask in a test, right? So we should find out. And this is a hinge. This hinge here connects the variable region, the FAB region, to the FC region. You go back here, this is the hinge, right? It's made up of disulfide bonds. So you get an idea of the types of amino acids that bind these 2 regions together. If you talk about disulfide bonds, what amino acid forms disulfide bond? The biochemist will be able to answer this. Yeah. So maybe this slide can take care of that later. For the history, I don't have to read this, you can read it on your own. All right, hybridoma technology, how monoclonal antibodies are formed. Right? It's a technique of producing hybrid cell lines called hybridomas by the fusion of a specific antibody producing lymphocyte B cell with a myeloma cell. A myeloma is a cancer cell, right, that has the ability to grow. Cancer cells grow continuously. They don't die. They are immortalized, right? So fusing an antibody producing cell to a cancer cell is a way of immortalizing the antibody producing cell so that it doesn't die. It's fused now to a cancer cell. Basically, that's the technique. So hybridoma produces antibodies. I'm sorry about the mistakes here, that have single specificity and called monoclonal antibodies. We already know what monoclonal antibodies are. So, all right. So this is the principle. Well, the principle of hybridoma is based on fusion between the myeloma cell, which is a cancer cell, and the spleen cell. The spleen cell contains the antibody-producing B lymphocytes, right? So you get that from the spleen cell. Of course, this has to come from an immunized animal, an animal that we've injected with an antigen of interest, specific antigen that will allow the production of a particular antibody you're interested in. Okay. Spleen can produce antibody but die in short period under culture condition because the spleen cell is not a cancer cell. So of course it has a lifespan, right? It's going to die at some point. Or by fusing with a cancer cell, so myeloma cells are adapted to grow permanently in a culture because their cancer cells immortalized, they keep proliferating. So in this case, you have a use for a cancer cell. Myeloma cells cannot synthesize antibodies as they lack this gene required for the synthesis of an enzyme called hypoxanthine guanine phosphorylosyl transferase. This, I think, is required in nucleotide metabolism. You can read about this if you want, and the other enzyme. So mutant myeloma cell cannot grow in this medium, hypoxanthine aminopterin thymidine. So the cancer cell alone, if it isn't fused to an antibody producing B lymphocyte from spleen cell cannot grow in this media. Or if you fuse a cancer cell, the myeloma cell, with the B antibody producing cell, they grow excellently. So you can use this medium for screening. Basically, that's what they're saying. If you produce your hybrid myeloma cell plus B cell, you use this medium to screen. The cancer cells that did not fuse with the B cell will not grow. B cell that did not fuse with the cancer cell will also die because they have a lifespan. It's the cancer cell that make them immortal, right? So only hybrid cell can grow in HAT medium and produce monoclonal antibodies. So you can read this if you want to understand more what I think I've said basically everything in the last slide. And then what immunization? Immunizer which through repeated injection of specific antigen for the production of specific antibody facilitated due to proliferation of the desired B cell. So produce tumors in a mouse or a rabbit. It's culture, of course, in culture the cancer, culture separate the spleen cell that produce specific antibodies and the myeloma cells that produce tumors. And then we fuse. The fusion stage. The fusion is done using polyethylene glycol. If you want to know more about this, you can browse videos on YouTube. It's production of hybridoma using PEG. So hybridomas are grown in this medium, which I've mentioned before. So they grow, if the hybridoma is formed, they grow well in this medium. If they are not formed, if the fusion is not successful, they are not going to grow. Basically, that's the principle behind the formation of hybridoma. After the formation of hybridoma, we prepare single colonies, subculture, things like that. If you have any lab experience, you know what I'm talking about. So culture selected hybridoma cells for the production of monoclonal antibody. After production of the hybridoma and screening, we make sure hybridoma produces the monoclonal antibody. You can now freeze your cells if you want to for future use, or if you want to apply it in that instant, can inject in the body of an animal, right? So that antibodies will be produced in the body fluid and recovered later, in the body fluid. So it's up to you, right? It's up to what you want to do. So. Okay, so monoclonal antibody solution. This summarizes everything I just said. Immunization, right? Immunize, antigen injected into the mouse. Isolation of antibody secreting plasma cells, right? And then fusion of the plasma cell to myeloma cancer cells, right? Look at this fusion here. So now this is done to immortalize the plasma cell. And then selection, then you have to screen them. After fusing, you see this myeloma cell has fused successfully, this one has fused successfully, this one has probably not fused, right? So if you screen, the one that has fused will produce antibodies. The one that has not fused will not produce anything. Look at the color of the medium, the medium, right? So plates in an 96 well plate, the ones that produce antibody probably have this color you're looking at, different color from the medium. And then maybe the ones, there's particular reaction, color change that will indicate that, yes, this is producing and this is not producing. Definitely, if you put the medium and the medium does not change, it means there's no production of anything there, right? So it's quite simple if you understand the principle. You know what I'm saying. So the screening is done with ELISA. What is ELISA? I think we should talk about ELISA, right? Direct ELISA. There are different types of ELISA, direct, indirect, sandwich, but here we just touched on direct ELISA. For direct ELISA, your sample now is added to the wells, right? And this is your sample. You've done the fusion, yeah? So of course, you're going to have a control where it's just the plasma cell, another control for myeloma cell, then another control for your fusion cells where you're hoping fusion is successful. And then you create 96 well plates, you run a reaction. So the antibody splitted in a well, right? And then conjugated antibody binds mobilized analytes. Enzyme aromatic color. Enzyme now use an enzyme to detect the presence of the production of that antibody. We design an experiment for that. So an enzyme-linked immunosorbent assay, is a test that detects and measures antibodies in your blood, right? This test can be used to determine if you have antibodies related to certain infectious conditions. The antibodies are proteins that your body produces in response to harmful substances called antigens. So I think that's it. It's hybridoma. And then there's another way of producing monoclonal antibody. It's called the recombinant monoclonal antibody production, where you use plasmids. We've done the recombinant DNA technology. The engineering aspect with laboratory. And I'm sure in other courses too, so you should know about this. This FD is the heavy chain of the FAB, the antigen binding fragments. So it has been inserted into this vector. And then we have the FC region, which we already know. FC is the crystallizable fragment, also inserted into this vector. The LC here is the light chain also of antibody. So depending on the antibody you're looking to produce, right? For mass produce, you co-transfect these antibodies into the plasmid and introduce them into a cell. Of course, you screen. After screening, you select cells that have taken up the plasmids and are producing your antibody. So now you can scale up, increase the volume, right? The volume, scale up, increase the output. And then you use a purification technique depending on what type of purification technique is suitable for your antibody. Protein G purification means most certainly they are using chromatographic technique here to purify the antibody. This is a chromatographic column, right? Wash, load, and quality control. And then you check if you have the marker for that antibody, and you run your sample to make sure that every chain and light chain coincide with that of the protein or antibody marker. It means your purification is successful, and you go from there, right? So basically, this is just a summary. If you want to get more information, you can research and read more about this. I think that will be it for this lecture. The next class, hopefully on Wednesday, I'll come and discuss the application of biotechnology in medicine and drug discovery, and then we'll have to talk about your continuous assessment after. I would say if you have any question, you can ask, but this is not a live interaction, so I'd ask you to drop any questions or comments in the comment section if you have. If you don't understand anything, you can always go online, right? This is very easy to grasp, even for those that don't have a background in maybe life science related courses and things like that. Thank you. It's fine.